Good evening, sir.
Next video screen
Sir, visible ಇದೆಯಾ sir?
ಇದೆ ಮಾಷೆ ಸ್ಕ್ರೀನು ಎನ್ಲಾರ್ಜ್ ಮಾಡಿದ್ದು
Enlarge ಇದೆ sir.
ಆಯ್ತು ಪ್ರೆಸೆಂಟ್ ಮಾಡಿ.
Good evening, everyone. Myself Sindhu. My topic is account of specialized transduction. Introduction. Transduction is a process by which DNA is transferred from one bacterium to another by a virus. It is also refers to the process whereby the foreign DNA is introduced into another cell via through the viral vector. Transduction is a method of gene transfer in a bacteria from the donor to recipient using the bacteriophage. In transduction, at first bacteriophage infect the donor bacteria and then carries some part of the donor genome with it. The DNA into recipient cell. And when go to the history of transduction, the transduction was first discovered in 1952 by the Jos Leidberg and the Norton Ginger. There are two types of the transduction. There are two types of the transduction, that is generalized transduction and specialized transduction. When moved to the generalized transduction, a DNA fragment is transferred from one bacterium to another by a lytic bacteriophage, which carrying the donor bacterial DNA due to an error. When come to the specialized transduction, a DNA fragment is transferred from one bacterium to another by a template bacteriophage, which carrying the donor DNA along with the phage genome. Due to an error in this phage, insert the genome at the specific site. When compared to generalized transduction, specialized transduction have a special characteristics. The process of transduction. When come to the generalized transduction, if all the fragment of the donor DNA from any region of the chromosome have a chance of enter to introducing the bacteriophage, then it is known as a generalized transduction. In generalized transduction, the first bacteriophage infect the donor cell and begin its lytic cycle. When a virus enter into the bacterial cell, the virus eject the host cell and synthesize the virus components such as the genome, enzymes, capsid, head, tail, and the tail fibers of the bacteriophages. Then the viral enzyme hydrolyze the host cell DNA into the small fragments. During the assembly of the virus component to the from of the progressive virus, sometimes any of the fragment of the donor DNA get incorporated into the virus capsid, that is the bacteriophage head. Such a abnormal bacteriophage when infect a new cell, it can transfer this donor DNA to new bacteria. Since this donor DNA is not viral DNA, but it does not replicate inside recipient bacteria, but it undergoes the homologous recombination with the recipient cell's chromosomal DNA, forming the recombinant cell. When come to the stages of the generalized transduction, the first stage is a phage first infect the donor bacterial cell. Then the infected bacterial cell come to phage of the DNA and the protein are made. And bacterial chromosome is broken into the pieces. Then the next step is the bacterial DNA enters into the phage DNA and the DNA is broken. Occasionally during phage assembly, the piece of the bacterial DNA are packed again in the phage capsid Then the donor cell lies and release the phage particles containing the each of the bacterial DNAs. When a DNA, bacterial DNA into the phage of carrying the bacterial DNA, it infects the new host cell, the recipient cell. Then the final step of the generalized transduction is recombination can occur and producing the recombinant cell with a genotype different from the both of the donor and the recipient cell. When come to the specialized transduction, when compared to the generalized transduction, specialized transduction is very special. In specialized transduction, bacteriophage transfers only few restricted genes, that is the DNA fragments, from the donor bacteria to recipient bacteria. The specialized transduction is carried only by the temperate bacteriophage, which undergo the lysogenic cycle in the donor cell. At the first, temperate bacteriophage enter into the donor bacteria, and then its genome gets integrated with the host cell DNA at a certain locations and remain dormant and pass generation to the generation into daughter cell during the cell division. The bacteriophage which follow the lysogenic cycle is also known as a temperate phage. In specialized transduction, when such lysogenic cells is exposed to certain stimulus, such as some chemicals, mainly UV lights, it can causes introduction of virus genome from the host cell genome and that begins lytic cell cycle. On introduction from the donor DNA, this phage genome sometimes carries a part of the bacterial DNA with it. The bacterial DNA lies on the sides of the integrated phage DNA are only carried during the introduction process. When such bacteriophages carries a part of the donor bacterial DNA which infect a new bacteria. It can transfer that donor DNA fragments into a new recipient cell. So in this specialized transduction, only those restricted gene are situated on the side of integrated viral genome and it have a chance to enter into the recipient cell. When come to the specialized transduction is commonly used for isolation and insertion of the genes of choice. For example, specialized transduction applications of specialized transduction. The specialized transduction is a phenomenon that occurs in a certain bacteria where the specific genes are transferred from one bacterium to another through the action of the temperate bacteriophage. It's a virus that infects the bacteria. This process can have several applications in a microbiology. The first application is gene mapping or genetic mapping. The specialized transduction can be used to map the location of the genes on a bacterial chromosome by observing by which genes are transferred by the phage during the specialized transduction. A researcher can deduce the order and relative the distance between those genes on the bacterial chromosome. When come to the gene transfer, the specialized transduction can be harnessed to transfer the specific gene to insert between the bacterial strains. This can aid in the genetic studies such as investigating the role of the particular gene in a bacterial physiology and metabolism or pathogenesis. When come to the gene function studies by transferring the specific genes into a recipient bacterium. The research can be investigate the functions of those genes in the new genetic context. This can help to educate the roles of various genes in a bacterial process. The specialized transduction can be used in a vaccine development. The specialized transduction can also use it in a engineer attenuated strains of the bacteria for use of the vaccines. Attenuated strains are weakened from the pathogen that can stimulate on the immune response without causing the disease. By deleting or modifying the virulence genes through the specialized transduction. The safer and more effective vaccine candidate can be developed through the specialized transduction. When come to the antibiotic resistance studies, the researcher can also use the specialized transduction to study the spread of antibiotic resistance genes among the bacterial populations.
ಅದು ಡಿಸ್ಕಷನ್ ಪಾರ್ಟ್ ಅದು ಎಲ್ಲ ಬರುತ್ತೆ.
This helps in understanding the mechanism of the antibiotic resistance disseminations and designing the strategies of the counteract it. Bioremediation. Some bacteria have the ability to degrade the harmful substances such as pollutants or the toxic chemicals through the specific enzyme encoded by the certain genes. Specialized transduction can be employed to introduce these genes into the other bacteria, effectively creating the strains that are capable to cleaning up environmental contaminants. When come to the industrial applications, the specialized transduction can facilitate the genetic engineering of the bacteria in industrial purpose, such as the production of enzymes, biofuels, and other valuable compounds. By transferring the specific gene into a well-characterized industrial strains, researchers can optimize the production of the process. When studying the regulatory networks, transferring the genes involved in the bacterial ensures that bacterial gene expression is controlled. This can provide the insights into cellular response to different environmental conditions. The specialized transduction can also use it in a investigation of pathogenesis. The role of specific gene in a bacterial pathogenesis by introducing or disturbing the virulence genes. Researchers can also assess this impact on the bacterium ability to cause of the diseases. When come to the evolutionary studies, the specialized transduction can also shed light on evolution of the bacterial genomes by revealing how genetic material has been transferred and rearranged over the time. Finally, overall specialized transduction offer a powerful tool for manipulating the bacterial genetics and studying of the gene functions and developing the practical application in a various fields of the microbiology. Thank you, sir.